Actually, when I teach the undergraduate or the master students, I always start with one question, and that's the same question I'm going to ask you. Who has ever smoked a joint? That's more response than I got yesterday. 40–50%. Wow, great. Then you might have experienced the feeling of being high, getting stoned, or a really relaxed feeling, sometimes getting hungry and going to the fridge in the middle of the night, having the munchies. All these effects of the principal active compound in marijuana, THC, are mediated through the cannabinoid CB1 receptor. And this is actually the most abundant GPCR in the brain. Here you see a picture of a rat brain, and all where it's red and yellow, that's the CB1 receptor. So it's expressed in almost all brain regions. And it's mediating the effects of feeling hungry or being relaxed. So, of course, this receptor is not there because we like to smoke a joint. This receptor is there because also the brain is able to produce its own marijuana. These endogenous substances, which can also activate the cannabinoid receptor, they are called endocannabinoids. And here you see the two structures of the most important endocannabinoids which can activate the same CB1 receptor. One is called anandamide, and the other one 2-AG, or 2-arginylglycerol. And these two endocannabinoids, they activate the CB1 receptor in all these different brain regions, and thereby causing also the same effects as THC from marijuana. Of course, we are not relaxed all the time, like I am now, but also we are not high all the time, so there is a mechanism how these compounds will be degraded, because the CB1 receptor is not always stimulated. Actually, in order to function for this system, these endocannabinoids, which activate the CB1 receptor, they have to be degraded. And for each endocannabinoid, there are special pathways to degrade these endogenous substances. So for anandamide, we have a special enzyme called fatty acid amide hydrolase, which cleaves this amide bond and which results in this fatty acid, arachidonic acid. Also for 2-arginylglycerol, there's a specialized enzyme called MAC lipase, and it degrades 2-AG also into arachidonic acid, thereby inactivating the function on the CB1 receptor, thereby stopping actually the activation of CB1. So the pharmaceutical industry has been interested in developing inhibitors for these enzymes, especially for the enzyme fatty acid amide hydrolase. Because if you can block this enzyme, which degrades anandamide, then you would expect to increase the levels of anandamide, thereby enhancing anandamide levels and activating the CB1 receptor. And this could lead to increase in appetite, so patients in need of food, like cachexia patients or AIDS patients, they need to have some stimulus for food intake. So that can be used, and also it's thought that if you experience pain, that if you inhibit this enzyme, you might experience an analgesic effect. And also fear is mediated through this system, so if you enhance the system, you would get an anxiolytic effect. Here you see the structure of the enzyme FA, and this can be used to specifically design drugs for this protein. And actually, this protein is a member of a really large family of proteins, the serine hydrolase family. And all these 200 different members of this family act through the same mechanism. They have a catalytic amino acid called the serine, which is responsible for the mechanism of action of these enzymes. So the pharmaceutical industry and also academic labs are interested in inhibiting this enzyme, and actually a lot of clinical trials were ongoing to target this enzyme. And unfortunately, until January of this year, everything went smoothly. But on the evening of Monday, January 11th, one of the volunteers who got an experimental drug, he experienced some headaches, and also blurred vision, and he was taken to the hospital the same evening. And then after a couple of hours, his situation worsened, and he was declared brain dead. So this person got 50 milligrams of this compound on the left, on the right. And this is the Fa inhibitor tested in that clinical trial. And this compound was tested in all preclinical toxicology studies in mice, in rats, in monkeys, in dogs, and there were no signs of toxicity, so this effect was completely unexpected. Actually, there was no good communication between the hospital and the clinical trial conductors, so on the next morning, 5 other volunteers received an additional dose of this compound. So they got 6 doses of 50 milligrams of this compound. This was the highest dose tested in these volunteers. And also those volunteers were brought to the hospital. And actually they found out that these patients suffered from symptoms related to a stroke. Until now, we don't know actually what happened. What is the molecular rationale for the cause of this death? So there were in all kinds of newspapers like in International, Volkskrant, but also CNN, Nature, they reported on this trial from what went really horribly wrong. And there's a lot of speculation what is the cause of this effect, but the short answer is we don't know. Actually, there's one main hypothesis, and that is that off-targets are responsible for this effect. Because other companies like Pfizer, Merck, Johnson & Johnson, they had also tested Fa inhibitors in phase 1 and phase 2 clinical trials. And these patients didn't suffer anything like the symptoms observed in this clinical trial. This suggests that Fa itself is not the main cause for these adverse effects of these neurotoxicity. Here you see actually the Pfizer compound. Piet Hein van der Graag, from the director from the LSCDI, he worked on this during his tenure at Pfizer. And this compound was safe in phase 2 clinical trials. So what's then the major hypothesis what went wrong is that the off-targets might be responsible. Because as I said, these compounds, they inactivate the enzyme by reacting with the nucleophilic serine in the enzyme, catalytic site. They attach covalently, and then this is found in the enzyme. So these enzymes are irreversibly covalently inactivated. So the big question is, how can we identify these off-targets? Because all these 200 family members of the serine hydrolase class could in principle react in the same way. So we have to identify which one of these serine hydrolases might play a role in that sense. And actually here our own research comes into play, because since a couple of years we are also interested in the serine hydrolase family. We are working on a different enzyme, but we use the same, we have developed different technologies to identify and to characterize the off-target activities of our own in-house developed inhibitors. And this we have published the last couple of years in JAX and Angola Chemie. And this method, activity-based protein profiling, is a very powerful technique to identify in a very efficient way the activity and the selectivity of these compounds in tissues. So how does it actually work? I will go a little bit into that, so bear with me, I will come back later to more simplistic view. So we have a general inhibitor, fluorophosphonates, which actually targets all the serine hydrolases. Sarin is also based on this principle, the nerve gas. And this reacts with almost all serine hydrolases. Then we attach a fluorescent reporter group to this general serine hydrolase inhibitor. And then we can, if we incubate this compound with a proteome or a tissue homogenate like brain, then we will be able to visualize all the different serine hydrolase activities in the brain. And this, if you put it on a gel, as the space gel, then you will get some kind of barcode on all these different red bands are corresponding to different proteins in the brain. Then if you incubate, pre-incubate your tissue homogenate with your inhibitor of interest, then if one of these enzymes is blocked by your inhibitor, then the probe cannot label this enzyme anymore, and this will be visualized on gel by an empty layer. So where the band is disappearing, this means that this protein has been inhibited by your compound. So in this way you get a very nice view of the activity and selectivity of your probe. So when we learned about this tragic accident, actually the next day, on Saturday, one of my PhD students, Ante Janssen, he came to the lab and started synthesizing this drug candidate of the trial. He made also some analogs of it, and the synthesis is about 7 or 8 steps and can be done within 2 weeks. So he prepared several analogs, and we sent these compounds to our collaborators in Italy, in Rome and Naples, because they have assays available to measure directly the activity of these compounds on the Fa enzyme. They have purified human Fa, and as you can see here, here are the activity plots of these compounds. We tested them at 3 different concentrations, and here you see the residual activity after incubation with these compounds. And compound 3 is the fatal drug. And as you can see here, it's actually the weakest active compound in this assay. So it's not a very potent Fa inhibitor at all. At 1 micromolar, then still more than 50% of the enzyme's activity is left. So that's not very potent. Whereas the comparators, like the Pfizer compound, they completely abolished the activity already at 1 micromolar. So that was already a big surprise. It's not a really potent Fa inhibitor. So next we wanted to test their selectivity. And for that we contacted the Netherlands Brain Bank, and they provided us with human brain samples from the frontal cortex and from the cerebellum. And that are these regions. So this is the frontal cortex, and here you have the cerebellum. So they provided us with 3 samples of each brain region from 3 healthy male donors. And we used that to test the selectivity using the ABBP method I just explained. So here you see the results of that profiling. So we have here the different barcodes actually, and I would like to focus your attention to panel C. This is this band here. This is the control sample. You see all different kinds of proteins being labeled by the probe, and then this band is the enzyme we are talking about, fatty acid amide hydrolase. So this is present in the different brain samples. And when we incubate them with the drug, which was used in the trial, then we see here that the band is disappearing. So indeed, this drug inhibits human Fa. As you can see here, it's actually quite selective, because most of the other bands, they don't disappear. If we look at another panel of enzymes here, also you see no bands disappearing. So, second surprise, it's already quite selective inhibitor as well. So we quantified, Annelot van Esbroek, PhD student also in my lab, performed all these experiments. And she quantified all the different bands, and as you can see here, actually, this is the Fa band, it's red, so most of it is inhibited by the different Fa compounds. And there is only one other band which is also inhibited. So this could be a potential off-target for this drug, the trial drug. And if you then look at this gel, you see here a clear band disappearing with the Fa inhibitor used in the trial. It's completely gone at 10 and 50 micromolar. So here we have an off-target of this enzyme. So now we have to identify what is the protein behind this band. And for that we used proteomics assay. We used mass spectrometry to identify the different peptides of this protein. And as you can see here, the results. We have one off-target identified, belonging to the family of the ADHDs. This is the only protein inhibited by the candidate used in the trial in France. Of course, we confirmed that Fa was inhibited, but all the other proteins which we could identify in the human brain are unaffected by this compound. And what's more important also, the compound which was safe in phase 2 clinical trials did not inhibit ABXT. So this could be a potential candidate to look further into. So we overexpressed this enzyme in the recombinant system. We used HEX cells and then overproduced this enzyme, and we had an activity assay using a substrate for this enzyme available, and then we measured the conversion of the substrate by this enzyme, and then tried to inhibit it with the 2 drugs. So drug 3 is the compound causing the neurotoxicity, compound 4 is the safe drug. Here you can see a clear difference in the activity of this protein, and we quantified it as an IC50 of 22 micromolar. And this is about 10 times higher concentration needed to inactivate this enzyme than for Fa, which is in line with the observed effects in this clinical trial. We further tested this compound on all different proteins related to the endocannabinoid system, and we could not find any effect on these different proteins. So within 2 months from reporting of this tragic accident, we have now performed the synthesis, obtained the selectivity profile in human brain, and so that's, I think, a very nice achievement for the PhD students who work day and night to obtain this. So to conclude, the compound tested in this clinical trial is actually quite weak inhibitor for Fa. It's remarkably selective, and despite all the speculation in the literature and the websites. And we have identified the first off-target of this compound with our chemical proteomics methods. And we have established that it doesn't interact with any other proteins of the endocannabinoid system. And when we look into the literature of the biological role of this protein, not much is known. It's a poorly characterized enzyme in the brain. So there is no toxicity data associated with this protein, so further research is required to see whether this is really the protein which is causing these side effects, but at least this is a lead into the further investigations. And another disclaimer is that perhaps there are other off-targets which we cannot detect with our methods, but that should also be further investigated. So I would like to thank my PhD students involved in this research, my international collaborators, MBO, and our faculty for funding of this research, because the endocannabinoid research is part of the profiling program, and the Netherlands Brain Bank for rapidly providing us with the human brain samples, and you for your attention.